Hi, welcome to week 5, chapter 5, photosynthesis. And I've noted here that we're also going to back up a little bit and talk about aerobic respiration again. That comes from chapter 4, and that will be at the end of chapter 5. So let's get started. Now we're going to get into the really good part of when we talk about energy. So if you go back to the first slide, we had talked about how light energy comes from the sun and it is taken up by plants, and plants will convert that light energy into chemical energy, which is done by photosynthesis. Well, this picture here shows a few different things. The reactants and the products at the end here. This is a chemical reaction for photosynthesis. So you have carbon dioxide plus water yielding or producing glucose molecule and oxygen. Okay, and just one quick note, if you look at the H2O where it says 6H2O, the 6 indicates that there are 6 water molecules, okay? It doesn't necessarily mean that there's 6 hydrogens and 6 oxygens, it just means that there's 6 water molecules. And same with the oxygen at the end, has 6 O2, and the 6 indicates that there are 6 oxygen molecules. Now in this particular depiction, the 2 next to the O should be in a subscript. It should be a little small number to the lower right of the O. So when we say O2, that's really how it's written, but it's not shown that way here. So here we have a photo showing what's kind of happening here. We've got the sun bringing in the sunlight, it hits the plant, and then we'll magnify that leaf to show you that there are these little cells inside the plant leaf called chloroplasts, which are the actual cell, and inside that cell is a component called chlorophyll, which is what's shown here. The chlorophyll is what gives the chloroplast its green color. Photosynthesis takes place inside the chloroplast. Okay, when photosynthesis is done, your product that's released is glucose and also oxygen. And so this is how we get oxygen in our environment. This is the oxygen that we breathe. If we were to get rid of all the plants in the world, we would suffocate because there would be no more oxygen. And so we would not have water, we wouldn't have any air to breathe, but this is where the oxygen comes from. So it's a byproduct of photosynthesis. If you'll notice, in order for photosynthesis to fully work and give off the glucose and the oxygen, we have to take in water and carbon dioxide into the chloroplast, into the chlorophyll area. Okay, and so think about when on your own person, when you inhale, you're inhaling oxygen. When you exhale, you're exhaling carbon dioxide, because that's the byproduct of the oxygen that your body's using. That carbon dioxide will go back into the plant. So the plant's giving you oxygen and you're giving the plant carbon dioxide. And so it becomes a cyclical process. This is one of the ways you can think of the circle of life, if you will. So the plants are giving off oxygen, we're taking in the oxygen, we're exhaling carbon dioxide, which is then in turn taken up by the plant so that the photosynthesis process can continue again and again and again. So I think this picture is a nice simple way to think about it. We've got the chemical formula here showing the reactants and the products. In this case, there's 2 products, glucose and oxygen. And then you've got the nice little visual picture of how that process happens. And think of it again in terms of breathing in and breathing out. When I was talking about light coming from the sun, light is a term that encompasses quite a lot. It's not just a beam of light, let's say. Obviously, the sun has many, many rays that we've seen before come from the sky. We've even seen on a really clear day, you can almost see all of the rays coming out of the clouds. We've seen that before. Those rays are visible light, and visible light is actually white. And you'll see here in this picture a rainbow, right? And then a horizontal rainbow here with some measurements next to it in nanometers. Visible light is white, but white light, it actually is comprised of all the colors of the rainbow. So if you're a kid, you probably learned the acronym ROY G BIV: red, orange, yellow, green, blue, indigo, violet. Sometimes we just say purple for short instead of violet. But that's the technical color, violet. Okay, and in this case it goes right to left, colors red to violet. When you smush all those colors together, you create white light or visible light. And this black bar here is showing you what a narrow scope of visible light there is coming from the sun. Now, also coming from the sun are all these other types of light rays, if you will. There's the gamma rays, which are the shortest wavelength, but the highest amount of energy. And then on that far side, on the right, are your radio waves, which are those long waves that we've seen before when we've talked about radio waves, and they have the lowest amount of energy. And so we've got this narrow window of visible light that comes to Earth, into our plants, that will be converted via photosynthesis into energy, chemical energy. And so again, you know, we had talked about the pH scale and how our body lives within a very narrow pH realm. Well, visible light that we can utilize to live is also a very narrow range. So our body really lives in a narrow window in all respects. And it's pretty astounding, because when you think about all the processes that the body goes through, especially as you learn about all the way down to these chemical processes and how abundant they are. I mean, billions of them happen in your body every day. When you think of it on that grand scale and how narrow a window that all of that happens, is pretty amazing. And I think that's incredible considering how complex we are. And so that's just one of those things that I think abstractly about, I suppose. Before we get really into the whole process of photosynthesis, I wanted to give you some terminology. And remember that the process of photosynthesis, your end products are going to be carbohydrates, which are basically turned into sugar or glucose, and those are stored in chemical bonds, or in and when those chemical bonds are broken, then the energy is released, right? And then the other byproduct is oxygen from photosynthesis. So this whole process happens inside the chloroplast. The chloroplast are the cells inside a plant leaf. Okay, so the chloroplast is the organelle of photosynthesis in plants and some protists. The chloroplast has this structure called the thylakoid membrane, and it has this folded like structure that's sort of stacked on top of one another, the folds that is, and they have there's a whole bunch of them basically that are connected to each other. So coming off of the chloroplast internal cell wall, you get this thylakoid membrane happening where some of photosynthesis happens. Okay, and then some of photosynthesis happens in the stroma, which is this semi-fluid liquid that happens between the thylakoid membrane, these stacked folds, and the chloroplast cell walls. And so basically is you've got this chloroplast, it's filled with stroma, and in that stroma you have the thylakoid membrane. Okay, and so I'll show you a picture of that next. In lab last week, or this week if you are in my other class, you were looking through the microscope to see chloroplasts. And so we took a plant leaf, we took a real thin layer of it, almost skin-like, and put it on a microscope slide. And when we looked through the microscope, we were able to see a picture much like the one at the top. So picture each one of those, see the little black outline, and then there's some white space, and then there's a whole bunch of little green circles within that black outline. Each one of those black outlines are a plant cell. Okay, the white part is the cytoplasm. That's that semi-fluid liquid that has some movement to it so that the chloroplast can move around and around within this cell. Now, if you were lucky enough, you were able to see that movement happen. And so you could see those chloroplasts moving along in a stream-like fashion inside the cell in the chloroplast, or excuse me, in the chlorophyll. I'm sorry, I said that wrong again. The chloroplasts are moving in a stream-like fashion inside the cytoplasm, which is the white part that you're seeing here. The green little circles, those are the chloroplasts. And if you look at the next picture, we've cut open a chloroplast, and when you peer inside, you can see those stacked layers, those discs forming from this cell wall, and those are the thylakoid membranes. Okay, and then the light green part is the stroma. Okay, so here's how it works. You have a cell, a plant cell. It has cytoplasm. Inside the cytoplasm, you have chloroplasts. When you break open a chloroplast or peer inside one, you have stroma, and inside that stroma are your thylakoid membrane system. Okay, so try not to get those confused. Think of it in that manner. Picture each one as you go. I kind of worked large and went inward, large to small in that respect of what I just said. Okay, so now we're going to get into photosynthesis. Real quickly, this once again is the chemical formula for photosynthesis. So we've got carbon dioxide and water mixing and using light energy from the sun to form a carbohydrate and oxygen. The best way to approach photosynthesis is in two components: light-dependent reactions and light-independent reactions. So if we break this down, light-dependent is the photo part of the word photosynthesis, and the light-independent reaction is the synthesis part of the word photosynthesis. I think it's best to do a lot of flashcards with these terms that I've been giving you, and with maybe some of the pictures I've been giving you. Perhaps put a picture on one side of a card, and on the back side put what that picture is. So you might put the chloroplast on one side, and then of course you'll write chloroplast on the other side. And again, I strongly urge you to gather study groups, whether it's one, two people studying or a whole group of people. But I think usually 4 people in a group is usually good. Sometimes 5 or 6 can be good. Usually anything more than that, then you have too many opinions and people talking at once and you don't get anything done. You find yourself ordering pizza. At least that's the way it happens in the dorm rooms that I was in. So light-dependent reactions convert light energy to chemical energy, which is just what we've been talking about this whole time. Light dependent, we're dependent on the light being the sun. So we're going to convert light energy to chemical energy. We're going to use ATP because we always use ATP for energy, right? And we're going to use this coenzyme called NADPH. All right? And at the end, we're going to release oxygen. So this is kind of showing you, hey, this is what, how we're going to make some of this energy and how we're going to make some of this oxygen, right? This whole process is going to occur in that thylakoid membrane that are in the chloroplasts. And you know what those are now because we just talked about it and I just showed you. That's that little ribbon looking thing that are all stacked on top of each other. That's the thylakoid membrane. And then the chloroplast is the little green circles. The light independent reactions are going to be using that ATP and the NADPH from the first part of the reaction, and it's going to make the glucose, which is going to be how we get our energy, how animals and organisms get their energy. They're going to use the form of glucose, right? And then other carbohydrates as well, because those are also can be converted to sugar. And it needs to have water and CO2 happening in this section of the photosynthesis. And then it's going to occur in the stroma. The stroma is that liquid part outside of the thylakoid membrane, but still within the chloroplast. Okay, so picture that. Go back and look at the picture if you need to. Go back and look at the chemical formula if you need to have an understanding of which parts we're talking about. Which side of the arrow are we talking about? Are we talking about the reactants or are we talking about the products? But these two components, light-dependent reactions, we're actually using the light that we get from the sun, and the light-independent reactions, we've already used light and now we're going to use the byproducts of that process and make our energy. Okay, and so those are the two that are involved in photosynthesis. This picture I really like because it gives you a really quick overview of the light-dependent reactions and the light-independent reactions involved in photosynthesis, and briefly the main players of photosynthesis. So on the left, we have the light-dependent reactions. Those are the ones that depend on the sun for light. So the light comes in as energy, and water also comes in. It'll be taken up by the roots and put into the cells. And once the light-dependent reactions are complete, water is booted out along what you see down at the bottom of the screen, the O2. But along with that, you'll see in the lighter green part in the center, the top line, NADPH and ATP are going to be released from the light-dependent reactions. And so they move over into the light-independent reactions. Those two properties are going to be now used in the light-independent reactions. Remember, these are the reactions that do not require the light. All right, we've already transferred light energy to chemical energy. Okay, So now we had the light independent reactions, and along with CO2 and water, we are going to produce glucose. And then if you also look, we're also producing NADP plus and ADP, which is moving back over to the light dependent reactions. So we have this little cycle that's going on. So I like this picture, just kind of gives you a brief overview of what's happening. And if you'll remember, the glucose in light independent reactions, that product will be used in aerobic respiration later. Now we're going to break down the steps in the light dependent reactions. There's quite a few of them. There's 8. Don't let them overwhelm you. I'm going to show you a picture on the next slide that will depict everything that's happening here. And so if you're able to get those slides side by side, like print them out, they'll make a lot more sense because the slide is actually numbered correlating to these numbers that you see on your screen here. So number 1, the light, let me back up first before we start with number 1. So what we have starting here is light from the sun is coming into the leaves of the plant, into those chloroplasts, right? Then we can start with number 1. Number 1, light energy will eject some electrons from a photosystem. All right, so we're going to eject some of these electrons, but they'll be used later, so hold on to those. Step 2, the photosystem will pull replacement electrons from the water, and that will release the oxygen that we saw in the previous slide, where the oxygen is sort of like a byproduct. All right, that's being kicked out. Electrons enter an electron transfer chain. So the electrons from step 1 are going to enter into this electron transfer chain, which is that ETC that we've talked about. This happens in the thylakoid membrane. And those electron transfer chains are used so that the electrons are actually going to work in a step fashion. So it's like they're walking downstairs, and you'll see this in the picture as they go down every step. So let's picture walking down a set of stairs and you're an electron. Every step you take downward, you're going to slow this reaction down. If you'll remember, when a reaction has energy happening, we've got these electrons moving around pretty fast. Reactions can start very quickly, and if we don't have a mechanism to slow them down, they could actually burn out the reactants before they've had a chance to make products at the end. So we'll either end up with the wrong product, or more likely we'll end up with no product. Okay, and all of that energy has been wasted. So electron transfer chain will slow down the energy so that the reactants have time to mix with the energy and make that final product that we're looking for. Okay, step number 4. Energy is used to form a hydrogen ion gradient across the thylakoid membrane. And again, you'll see this in the picture. We're going to have more hydrogen on one side than the other, and so typically there'll be hydrogen excess inside the thylakoid membrane and less outside. So if you think about the concentration gradient, high moves to low, so these hydrogen are going to move from the interior where there's an abundance of them to the exterior where there's fewer of them, to make that homeostasis balance that we always look for. Okay, so the hydrogen, that's how they get out. All right, the other photosystem, or another photosystem, will receive electrons from the electron transfer chain, and they're going to repeat this process again. So they're going to have water and light energy and the hydrogens, and then another electron transport chain, and all of that second phase, even though it's a repeat, that second phase is going to make this coenzyme NADPH, and that's what's formed. That gets thrown over to the light dependent reaction that we'll get to in just a little bit. Number 6, electrons will move through that second electron transport chain. Oh, and I just repeated that. And NADPH will be formed. All right, so that only happens once there. Number 7, moving on, the hydrogen ions again will flow across the thylakoid membrane through an ATP synthase. And so it's sort of like this little helper molecule that's going to take those hydrogen ions, and along with the energy of ATP, we're going to move through the thylakoid membrane out into the cytoplasm, and then we're going to move across over to the dependent reaction. Okay, and then the ATP will form in the stroma. All right. Then we have this electron transfer phosphorylation. I'll just give you a quick brief definition. It's the metabolic pathway in which electron flow through electron transfer chain sets and a hydrogen ion gradient that will drive ATP formation. So basically phosphorylation means that you are losing one phosphate group. So if you're talking about ATP, that's triphosphate. That means it has 3 phosphate groups. If we're taking away one, which often happens, we're left with ADP, which is diphosphate, or that means 2. Okay, so we've got, we're going to take away one at least, and we're going to be left with some, in this case 2. Now let's move to the next slide, and hopefully all of this will make sense. So this picture is not in your book, but I find that it's a better picture than what your book actually talks about. This is the light-dependent reaction. Okay, so it's showing you in the upper left corner right under the title, there's these little green stacked things. Those are your thylakoids. And so it's showing you that light-dependent reactions take place in the thylakoid membrane. Okay, and here you can see the lipid bilayer on the outside, and then on the inside, that green oval racetrack looking thing is the thylakoid compartment where a lot of this stuff happens. And so let's go through this picture. Oh, one more thing I wanted to point out. If you'll notice underneath this whole picture, there's a word called stroma. So all that white space that you see, just picture that as the interior of the chloroplast. And so when you are outside of a thylakoid compartment, you are in the stroma of the chloroplast. Okay, remember chloroplasts sit in cytoplasm, but the interior of a chloroplast is stroma and then has thylakoid compartments. Okay, moving on. Number 1, obviously light energy is coming into the chloroplast, into the thylakoid compartment. Water was also taken up, and at this point it's going to be broken up. Hydrogen ions, you'll see, go to the right, and oxygen ions leave the thylakoid compartment, go into the stroma. And then the electrons will enter in the electron transfer chain, which we just talked about. Then you'll see at step 4, it's going to repeat itself. Step 4 is showing how all these hydrogen ions were formed in addition to taking on the 2 from the water breaking up. And it's going to start this electron transfer chain again, and it's still taking in light at this point. And so we repeat that electron transfer chain. At the end of this second round, we get the coenzyme NADPH. All those hydrogen ions that we see at the end of the second electron transfer chain, or excuse me, at the end of the first electron transport chain, all those hydrogen ions move all the way down to number 7 to the right there. They are going to go across that hydrogen gradient. You can see there's only 1 on the outside of the compartment in the stroma, and so it's going to drive all of those other hydrogens across that gradient using that ATP synthase, which is just a little channel that opens up and uses ATP to do that, and we get this balance of hydrogen. Then the NADPH will also leave the compartment, and you'll see here that the ATP has lost a phosphate group, so now we're calling it ADP plus the phosphate, and they're going to move downward into the light-independent reactions. So really quickly, light comes in, water's broken up, oxygen's released, hydrogen moves down the compartment, electron transfer chain happens, more hydrogen ions are produced and moved downward to the right. Then it repeats with the light energy and electron transfer chain. We produce NADPH, and NADPH along with hydrogen are going to cross over into the stroma, and they're going to move over down towards the light independent reaction. At the same time, ATP is breaking up because we're utilizing energy, and we're left with ADP and a phosphate group. So that was like the really fast version of what we just did. So this slide is just giving you a quick overview of the light independent reactions. So now we are independent of light. We don't need them anymore. We've used up the light that we had, and we enter in what's called the Kelvin-Benson cycle. And so we are taking that NADPH and the ATP from the light dependent phase, and we're entering into this Kelvin-Benson cycle. I think your book actually just calls it Kelvin cycle. For some reason they've dropped the Benson. I don't know why. When I was a kid we called it Kelvin-Benson cycle. So whatever you want to call it, either one is fine. Again, these are the light independent reactions of photosynthesis. We're taking the NADPH and the ATP from the dependent phase, moving it into the light independent phase, and we're going to work with cyclic carbon fixing pathway that will form the sugars from the carbon dioxide. Okay. It will be driven by ATP, of course, which is energy, and the electrons that came with the NADPH. During this process, the main thing to know is that it's a carbon fixation process, which means that carbon from an inorganic source, such as carbon dioxide, will get incorporated into an organic molecule. And we'll see how that happens in just a minute. There's 3 stages to the Kelvin cycle. Stage 1, the enzyme called Rubisco incorporates into the CO2 and into the organic molecule. So actually it's taking the CO2, utilizing it, and it's all done inside an organic molecule. Stage 2, the organic molecule from stage 1 is going to be broken down even smaller. And stage 3, this molecule RUBP, I guess there's no way to really say that well, but in an acronym form, but RUBP is a molecule that will start this whole Kelvin-Benson cycle, and it's regenerated again and again so that the cycle can continue. And this cycle can repeat billions of times. I mean, it's really not going to wear out too quickly. All right. So now let's take a look at the next slide and see how this actually works in a picture form. So this slide is showing you again the chloroplast is this whole unit that you see, and then the inside, that light green area, is the stroma, and then it shows you the little thylakoid compartments there, those green disc shapes all stacked up, are included in the stroma. But we're going to hang out in the stroma for this. So you can see on the left hand side we've taken the NADPH and the ATP, and we're going to enter them into this Kelvin-Benson cycle. At the same time, we're taking in CO2 that comes into the chloroplast from the outside of the plant. Remember, CO2 is what we breathe out, and so that CO2 it will be taken up by the plant, and it enters into this Kelvin-Benson cycle as well. As it does so, it will convert this 3-carbon compound called a PGA or a 3-PGA into another carbon compound called the G3P. And it doesn't say that on this slide, but that's how it works. And that is called a reduction reaction because it's going to gain electrons. And the reduction is the gain of an electron by an atom or a molecule. In this case, it's ADP, remember D, and NAD plus that's happening. So I'm just giving you some background that's not on this slide. Okay, the resulting reduction reaction is going to return to the light dependent phase once it's energized. All right, so now we're moving into the Calvin cycle some more. Again, we've taken that CO2, the ATP, NADPH, we've converted this PGA, and we go around and around and around. And as we do, some glucose will be spit out, but before that happens, it'll go around, and we're going to pick up some more energy on the right, that ATP, and we're going to pick up this other molecule called the RUBP. All right, and the RUBP stands for ribulose biphosphate. It's got 5 atoms of carbon and a phosphate group on each end. So depending on what it looks like, which we don't have depicted here, but it will enter the system and enable it to prepare for the carbon fixation stage. And it's also going to use that ATP, like I said. All right. And so as this goes around, we took that Rubisco and the carbon dioxide and the NADPH and the ATP. We run around, we use more ATP. We've got this RUBP happening. We've got that reduction happening with the carbon. Now we can start the fixation cycle. At the end, every turn this Benson cycle makes, it will fix carbon atoms from the CO2. And so it will take 6 turns of the Calvin cycle to fix 6 carbon atoms from the CO2. And then those 6 will require energy input from 12 ATP molecules and 12 NADPH molecules in the reduction step, and then 6 ATP molecules in the regeneration step. It has to do that all around in order to produce one glucose molecule. So let me say that again. The Calvin-Benson cycle or Calvin cycle will have to make 6 turns or 6 circulations in order to produce one glucose molecule. So you can see how it requires a lot of energy to get this Calvin cycle started and to keep it in motion to get that glucose that we need. And then the glucose is still going to be in the stroma until we need it. Here's a nice way to think about what we've just talked about. We've thrown a lot of information at you. Aerobic respiration starts in the cytoplasm, and it's called glycolysis, which is stage 1. There are reactions in which glucose or another sugar is broken down into 2 pyruvates and will release 2 ATP molecules. Coenzymes, in this case 2 NADH molecules or enzymes, will pick up the electrons and then transfer them into the citric acid cycle or the Krebs cycle, however you want to think about that. Those electrons are moved into the mitochondria, and that's where this citric acid cycle takes place. So we've got acetyl-CoA formation that's come from the glycolysis stage, from the pyruvates basically in the glycolysis stage, and we've formed this acetyl-CoA, and then we've broken down pyruvate to carbon dioxide. We've netted 2 more ATP, and we've reduced coenzymes. Now we have 8 NADH and 2 FADH2. There's a note here that says the text on page 90 incorrectly shows 6 NADH. That is actually for a different textbook, not the one that you're working on, not the textbook called Concepts of Biology for the Bio 156 class. So just ignore that little note. There are 8 NADH and 2 FADH coenzymes produced, okay? And all of this occurs in the mitochondria. So glycolysis occurs in the cytoplasm, and it starts with glucose. It ends up with 2 pyruvate, and then the pyruvate along with the electrons are going to cross into the mitochondria and start the citric acid cycle, which will take the pyruvate and utilize an electron transport chain to get the acetyl-CoA formation, and you're going to be giving off CO2. You're going to be gaining two forms of energy in the form of ATP, excuse me, and then you're going to have these coenzymes as well. And again, that occurs in the mitochondria. So these are the most important facts about aerobic respiration that I can share with you. So basically, glycolysis is sort of a byproduct of photosynthesis. You can see here at the top we've got this 6-ringed glucose molecule, and through some energy happening here, the glucose molecule will break apart basically and be utilized, releasing energy, and the energy again is in the form of ATP, and here we're releasing 2 ATP molecules, and then some ADP have been produced as well. Once that energy, the ATP and the ADP have been taken apart or taken out of this glucose ring, you're left with this stuff called fructose diphosphate. Remember, now we've got 2 phosphate molecules here. Each one of those will break up into these, it's called the glyceraldehyde triphosphate or 3-phosphate, and you'll have 2 of these, and now you can see they're on very separate paths. So you've got the left hand side and the right hand side, but each side are producing the same things. Once it goes through these chemical processes using coenzymes and cofactors, you've got NAD plus NADH being extracted. You've got more ADP and more ATP being extracted, and then your end product is what's called the pyruvate. Okay, and then you've got each side giving off both of those, right? And the energy and the coenzymes. So basically, if you count up the ATP, we've got essentially 4 being produced by glycolysis. So we've got 4 energy molecules being produced. Here's a photo depiction of aerobic respiration. So the previous slide showed how we ended up with 2 columns, and at the end of those 2 columns were pyruvate. Well, the pyruvate ended up coming into the citric acid cycle as pyruvic acid, because there's 3 of them together. Along the way, it's going to pick up a coenzyme A and an NAD+. The coenzyme A will spit out some carbon dioxide. The NAD+ will spit out some NADH. In turn, when that's completed, we have 2 carbon molecules and an acetyl-CoA molecule. Sorry, I'm having trouble saying that. Let me back up. We have 2 carbon molecules and a coenzyme A. Together they make the acetyl-CoA enzyme. All right. As that enters the citric acid cycle, it spits back out that coenzyme A, and it can be used again and again. Along with all of these parts, so we've got the carbon coming in, we also have 3 NAD+ molecules spitting out 3 NADH molecules. We have 1 FAD molecule spitting out FADH2 molecules, and then 2 carbon molecules are going to be released in the form of carbon dioxide. So we're getting rid of carbon dioxide there. Okay. At the end, at the bottom down there, ADP comes into the citric acid cycle and makes ATP. So the citric acid cycle has to make a couple of turns in order to get these two carbon dioxide molecules. It's going to take 6 carbon atoms from the original glucose that we have and will eventually release as carbon dioxide. It takes 2 turns of the citric acid cycle to produce the equivalent of 1 glucose molecule. So it's pretty interesting. There's a lot of energy happening here, and carbon dioxide being spit out. The citric acid cycle, you may have heard before, it's also called the Krebs cycle. But this textbook calls it the citric acid cycle. And so basically what you're ending up with is 6 carbon dioxide molecules at the very end of the citric acid cycle. This whole citric acid cycle happens within the mitochondria. That's very important. I'll say it again. The citric acid cycle or Krebs cycle happens inside the mitochondria and will produce 6 carbon dioxide molecules, whereas the glycolysis happened in the cytoplasm. It started with glucose and then it ended with those 2 pyruvates, okay, which were then entered into this whole citric acid cycle thing. All right, so that is aerobic respiration. Very different than the photosynthesis. Some of the same moving parts, but utilized in a very different way. Here's a picture I really like. It's not in your textbook, but I think it gives a really nice visual of what's happening in the aerobic respiration process. So if you look at this picture, the top part shows you where glycolysis happens. If you'll notice the white area, if you will, picture that to be the cytoplasm inside the plant cell, right? All right, so we've got the cytoplasm. We have 2 molecules of ATP and 2 NAD plus entering into, along with glucose, this glycolysis process. Now remember, glycolysis literally means the breaking down of glucose. So we're going to take that glucose that we got from photosynthesis, and we are going to take out some of the coenzymes. We've gone from the NAD plus to the NADH, and then we have 2 pyruvate, and then we've got a net yield of 2 ATP happening off to the side there. Then the NAD plus will enter into the mitochondria as well as the pyruvate. The NAD2 is going to meet up with the pyruvate. It's going to have 2 CO2 molecules removed, and you're going to have a product of 2 acetyl-CoA molecules. Those are going to enter the Krebs cycle along with we now have 8 NADH and 2 FADH2. This is where it should say 8 and not 6 NADH. So we have 8 NADH and 2 FADH2 entering into the Krebs cycle along with the acetyl-CoA. As we go through this process, we are going to get rid of 4 CO2 molecules. We're going to have those released, and 2 more ATP molecules. And so this whole process thus far has given us a total, along with the electron transfer phosphorylation happening, 32 ATP molecules. So we've just from 6 glucose molecules, we've netted 32 ATP molecules. That is a lot of energy, and I want you to remember that when we start talking about anaerobic respiration. Okay, and note the difference in how much ATP is being net in this case. All right. And then the next slide will explain this electron transfer chain. The word phosphorylation just means that a phosphate group is being removed or added to another phosphate group. Okay. So here we have another picture that is just being carried on. This would actually be attached to the previous picture off to the right of it. And so we kind of went from a north to south realm, from glycolysis to citric acid cycle. And now we're going to the right and entering into the final stage of aerobic respiration. And this is where the electron transfer chain that I talked about earlier happens. And again, we're utilizing hydrogen ions and these coenzymes, the NADH and the FADH2, and we have to have these electrons entering in this chain to lower the amount of energy happening so that we can have the end product of producing the ATP and all the hydrogen, and then of course the water as well. And so what this does is, once you go down this stair step of electron transfer chain, you will take the oxygen that's formed and you'll grab onto some more hydrogen that's in this membrane, and some of it will shoot across the membrane back into the cytoplasm, and the energy, the ATP, is going to be yielded and to be used later on for other processes. And that ATP also comes from the addition of the ADP and the phosphate that it picks up along the way as it crosses over. But the biggest part about this final stage is that it has an electron transfer chain that slows down the energetic process so that we can get these end products as we expect and need, and not burn up too quickly. This is just a quick little depiction of aerobic respiration to show you how it interacts with photosynthesis. So photosynthesis is at the top in the green arrow. Notice the energy coming in. That would be called an endergonic reaction. It spits out glucose and oxygen. Both of those will enter into aerobic respiration, which includes the glycolysis using the glucose and the citric acid cycle, and which produces this carbon dioxide and this water. It will also spit out, look at the bottom, it'll spit out energy, and that is called your exergonic reaction. So energy in and energy out. Okay, what I want you to notice is if we had the photosynthesis chemical equation up here, it would show that the photosynthesis will pretty much go left to right, and then you can reverse that and go right to left, and that's your aerobic respiration chemical formula. Okay, so you can turn them around, forward and backward, photosynthesis to aerobic respiration, or vice versa, depending on where you start. Okay, so this is just a nice little picture I like that kind of concisely sums up everything we were talking about.